So in the prior videos on science methods related to designing a controlled experiment, we learned how to formulate a scientific question, how to write hypotheses, how to design or what are the components of a controlled experiment, and then how to handle variation in within an experimental group. And that was calculating the standard deviation. Now in this video, we're going to go over how can you use your calculation of mean and standard deviation for your controlled and your experimental group. How can you use both the mean and the standard deviation to figure out or determine if the two groups are significantly different or not? In other words, you're going to learn how to make an objective determination as to whether or not your test variable caused a significant difference in the response variable between the two groups. In the context of the corn plant experiment, whether or not removing magnesium or not caused a significant effect on the growth. And so we have to look at the means and the standard deviations. And one of the main reasons here is you don't really know what the true mean is. We have an estimate of the mean from the experiment. And in this case, the variation is important. Now, this is taken from page 5 of your lab manual, and it's in the context of the yeast experiment where the yeast are exposed to different nutrients and how much carbon dioxide they give off. So that experiment had its own controls, and then different nutrients were given. Those were your treatments. So they measured, they did replicates, and they came up with mean and standard deviation for each treatment. And the authors of the manual suggest a way, an objective way for determining significance. In actual statistics, there's a significance test, an inferential statistical test that assigns probabilities as to whether or not your means are different or not. Doing that very formal way is beyond the scope of this course. So hopefully you will at some point be required to take a statistics course. It's a tool used by scientists in every science field. It's used in other fields as well. It's used in sociology and political science and so on. So statistical literacy is important. But we're going to use a sort of a quick and dirty method for making an objective determination. And so the author suggests that for us, instead of going through and learning how to do that probability inferential test, we're going to use this, and it's right here. It says we're going to make a judgment as to whether or not the two means are significant if the means, remember you had to calculate a mean for each group, right? So if the means plus or minus 1/2 of the standard deviation do not overlap. So basically what we're going to be doing is we're going to take in the mean for your control group, and we're going to add and subtract it, not a standard deviation, but we're going to take half of that standard deviation, okay? And that's going to give us a range because we're going to get the mean and we're going to add half the standard deviation, and then we're going to get the mean and subtract half a standard deviation, and that's going to give a high and low value, okay? You're going to also do that for your experimental group. And you're going to add, the experimental group is going to have its own mean. You're going to add 1/2 and subtract 1/2 the standard deviation, and that's going to give you a high and low value. So in your high and low values, whether or not they overlap, if they don't overlap, then the means are significantly different. And the reason we have to do this is because the means are always going to be different, but we're trying to give an objective way of saying they're really different or they're not that different. And so if they're not that different, then we would accept the null hypothesis. And they're going to be judged to not be that different if the ranges overlap. If they don't overlap, the two ranges that we calculate with these formulas here based on this criterion, if they don't overlap, then the null hypothesis will be rejected and we accept the alternate. So I have here, instead of going and calculating the mean and standard deviations for two different data sets, This is a different context here. Here they're testing some variable, we don't even know what it is, and then they're looking at the growth of seedlings. These are little baby or young plants that have started to germinate and grow from seed. And they're looking at the shoot lengths. The shoot is the structure that grows above ground, the roots and the shoots. So they're just measuring the shoots, the part above the root that would normally grow above ground. Now, just taking a look at this table here, this is a table of your descriptive statistics, what you should always report when you're reporting your research, when you're reporting the results of your scientific investigation, your experiment, your study. You can see there's two groups, Group A and Group B. And let's just make up that Group A is your control. And we're just going to call it Group A. And Group B is your experimental group. Okay. If you look at the first column here after the groups, the two different groups, N is for the number of replicates. So there was 5 replicates for each group. This right here is your mean. I forgot to write mean there. It's your mean length in millimeters because they're looking at seedling lengths. And the mean for Group A is 10.1 and the mean for Group B is 19.0. And this last column is the standard deviation for the 5 replicates of 6.2. So it's the standard deviation of the mean, 6.2 millimeters and 8.4 millimeters for this one here. So the mean and the standard deviation for the two groups, now this is a different context, different experiment, but the same would be done for the magnesium experiment and the same will be done for the experiment you designed. You're going to have at least 2 groups, and that means 2 means and 2 standard deviations, 1 for each group. Okay. Now, the means are different, and it looks like the experimental group, the growth of the seedlings for whatever treatment variable that was, and we don't know what it was. I'm just saying some variable they're testing. Okay. It looks like when they tested that, they had exposed the Group B to it, because that's the experimental group. It seems to have had an increased effect on the growth because we have 19 millimeters after the experiment was done, whereas the control was only 10. Maybe they gave it fertilizer or whatever, I don't know. Fertilizer is plant food, right? And then these are the standard deviations. So they've already calculated that for us. They already went through the process of going through this table we went through in the last video to calculate your mean and your standard deviation, something you need to practice. And now we're going to take those means and standard deviations, which is the next step, and do our significance test. And the significance test is to determine if the mean plus or minus 1/2 standard deviation don't overlap. If they don't overlap, then that's going to be considered significantly different. There's a significant effect. Okay. So what would our hypothesis be? First, it helps to know that, right? Because if the ranges don't overlap, then the alternate hypothesis would be that whatever the test variable is, maybe it's fertilizer that they gave, is going to affect the growth of the seedlings. The null hypothesis, which actually comes first, says that the test variable has no effect on the growth of the seedlings, right? Notice the difference in the usage, effect and affect. Effect, the way it's written here, an effect is a noun, affect is an adverb, okay, or a verb. So that's why it's the 2 different words. There's a difference between the 2. But basically, the alternate says there's going to be an effect of the treatment, and the null says no effect when you do the treatment, right? So the alternate, the null says if I give fertilizer or whatever to the plants, that's going to have an effect. Not going to have an effect. We're not going to get a difference in the 2 groups. And the alternate is the same, the opposite. It's going to have an effect. So now we need to see, does this experiment, the data, support or reject the null hypothesis? So what do we need to do? We need to apply that standard, the significance standard. So what we need to do is, for Group A, our control, we need to take the mean for that group, and we need to both add and then subtract 1/2 the standard deviation. We're also going to need to do that for Group B. We're going to need to take the mean, and we're going to add 1/2 the standard deviation, which is calculated by the methods we did in the prior video for both. So the information to do that is in the table. They already calculated the mean and the standard deviation, something you'll have to do with a raw data set from an experiment. So looking at these values here, let's go ahead and use this formula to guide us. The mean for Group A is 10.1, and in the first case, we're supposed to add 1/2 the standard deviation, 1/2 of 6.2. And that is going to give us a high value for the range. And then we also now need to subtract. So we're going to subtract 1/2 the standard deviation, which is 6.2. And then that's going to give us the low value. So this will give us the high value for the range and the low value. Let's do this. Go ahead and set it up for group B. Group B, the mean is 19.0. And we need to add 1/2 its standard deviation. The standard deviation for group B is 8.4. And that's going to give you the high value. Then for the low value, we're going to subtract 1/2 the standard deviation, which is 8.4, from that mean. Okay, so remember the order of operations. You always multiply and divide first before you add. So we need to do the 1/2 times the 6.2. When you multiply by 1/2, that's dividing by 2. So that really gives us a value of 3.1. So this right here would be 3.1, and that would also be 3.1. So really what we're going to do is we're going to take 10.1 and add it to that quantity. The quantity of 1/2 times 6.2 is 3.1. So 10.1 plus 3.1 is 13.2. That would be your high value above the mean. Okay, it's 1/2 the unit, the standard deviation units above the mean. The second one we're going to subtract, and the answer to 1/2 times 6.2 is still 3.1, but we're subtracting 3.1. So 0.1 minus 0.1 is 0.0, and then the 3 should be 7, 7.0. 9, 10, 10. Yeah. Okay, so we're going to do the same down here. So this would be the low value for the range. The range for A. So this is the plus or minus 1/2 the standard deviation. That's the range for group A. For group B, we're going to have to do that calculation. Now 1/2 times 8.4 gives me half of 8.4 is 4.1. So I'm going to add 4.1 to 19.0. 19.0 plus 4.1 is going to be 23.1. And then 1/2 times, again, is 4.1, but we're going to subtract that from 19.0. So that's going to give me, it's not 4.1, it's 4.2. Half of 8.4 is 4.2, not 4.1. 4.2 plus 4.2 is 8.4. Okay, so this should be 0.2, 23.2. And then again, 4.2 we're going to subtract. So when I borrow, so 19.0 minus 4.2, I got to borrow 1 from here. It's going to be 8, 4, so it would be 14.8, about 15. Okay, so the idea here is this is your high and low value. This is your plus or minus 1/2 standard deviation. This is your plus or minus 1/2 standard deviation. When I look at these here, the highest value here, which is 13.2, does not reach the lowest value of the other one. So right now I can already tell there's no overlap, and that would tell me that there is a significant difference. But let's take a look at how that looks in a bar graph. We're going to always graph, guys. You always graph your response variable on the y-axis. In this case, the response is your dependent variable, the length. The test variable, or your independent variable, which is your treatment. Fertilizer or no fertilizer, magnesium or no magnesium. Going back to the example with the tadpoles in that practice assignment from the prior video. Meat diet or lettuce diet, that's always on the x-axis. So here, this is your experimental groups. We have a group A and we have a group B, and then you always label your axes like we did here. This is your groups, which are your treatments here. You could say control and experimental if you want it down there. And here your variable here is the length of the shoots, so the seedling shoot length. So here is shoot length, and always put the units. Okay. Now I'm going to go ahead and label this 0, so that would be 0 millimeters. The units are millimeters. Okay. And I need to fit up to a high value of 23. So if I go by, every little line is ones. That's there's 5 there. That's 5, 10. I think that's going to fit my data because I won't go off the graph here. 15, that's 20. 25, that's 30. We should fit 20, the highest value that we need to graph there easily. Let's go ahead and graph the means first. The mean for group A is 10, about 10, 10.1. So This line is 10, the next one is 11, so 10.1 is just barely above the line, so it's right around the line, just above it, not much higher, because it's 10.1, not 11. So that's, the bar is the mean for group A. The mean for group B is 19.0, so this dark line up here, that's 20, so the little line right below it, that's 19, so this would be 19 right here. And that bar represents my mean. And normally, if we were in a middle school science class, we would say, oh, look, there is a difference right away and everything. But now we're at a higher level, and we're going to say, are the means really different or not? Because we have to compare the variation in these samples here. And that variation is going to be compared by whether or not the range of plus or minus 1 standard deviation overlap or not. And that's going to be easier to see if we graph it. So let's go ahead and graph the high value and the low value as a bar within the bar graph. And the high value for the first group should be 13. So if this is 10, 11, 12, 13, that's going to go to about right there. It's supposed to be 13.2, so really just a little above there. And that's the high mark. Remember, the bar itself is the mean. And then we got to go below to graph the low value of 7. So if that's 10, 9, 8, 7. 7 is about right there. And that's your low value. So this is your mean. Okay. That's X bar, the mean. The top of that error bar, we're going to call that an error bar, is X bar plus 1/2, the standard deviation. The bottom is the mean minus 1/2, the standard deviation, which is what we're comparing. Okay, so we're going to do that for the second one. And while the means are different, we want to see if the error bars overlap or not. So the high value for the error bar in the second one is 23.2. So this is 20, 21, 22, 23, about right there, just a little above it. This is not that fine of a scale for finding exactly where the 0.2 is. It's closer to 23 than it is to 24, and it's just a little above it. And then we got to go down to 14.8, which is near just below 15. So this line right here would be 15, so it's really just a little below it, almost, because it's 0.8. Come on now. And so that's your error bar for the second one. Now, if I draw a line, a dotted line coming all the way this way here, you can see that that low part does not overlap with the high part of the next one. So the two error bars do not overlap. And according to what it says here, if the mean plus or minus 1/2 standard deviation do not overlap, then we say that the means are significantly different. So in this case, the means are different. They're not just different, they're significantly different because the error bars didn't overlap. So that's our significance test. Simply looking at the mean plus or minus the 1/2 standard deviation, which are the error bars, the high and the low value here, and determining if they don't overlap, then there's a significant difference. So what do these results suggest? The results suggest that there was an effect with whatever they treated it with. Remember, I didn't really identify the test variable. Maybe they gave fertilizer to group B. I don't know. I'm just, this is a generic example using the growth of seedlings. Okay. Because the error bars don't overlap, there was an effect. So that means we reject the null because there was a difference. Remember, the null says no effect. So we reject the null and we accept the alternate hypothesis. Okay. So now we know how we've gone through the test. Now let's say that we ran a third group. Maybe we tested a different brand of fertilizer and the mean is bigger. So this is just a hypothetical adding. We had a third group. We had a third group here on the table, group C, and a different treatment, maybe a different brand of fertilizer. And it gave a bit more growth here. The mean is higher. It looks like it's about, it's just below the 15, about 14, and that is higher than the 10 for 10.1 for the first group. But we got to see are the error bars. And let's say that the error bars, when we went in, we calculated the plus or minus 1/2 standard deviation. we're skipping the step of doing all the calculations. Just as a hypothetical example. The whole thing is a hypothetical example. But our error bars ended up something like this. Okay. And so it looks to me like these two error bars overlap. They overlap right here in this region. And that tells me that B and C are not significantly different. Okay. But B and A are significantly different. Okay, but what about when we compare the control A to that other brand of fertilizer, if fertilizer was what was being used? Is fertilizer C giving us a significantly greater effect or different effect than the other one? Let's take a look at the low line here. I'm going to draw it coming all the way this way. And we see that it overlaps with A. And so group C and A would not be significantly different. And in that case, the null hypothesis would not be rejected if we were comparing group A and C. But when we compare group A and B, the null hypothesis is rejected. So in some cases, you have more than one treatment, and you have to consider each one individually as you compare it to the control. Okay, so that's how we're going to do the significance test, and hope this helps as part of what you're going to be doing when you run an experiment. Okay, this is how you're going to determine if your groups, if the treatment is significantly different from the control.